In this tutorial, we are going to explain how the Mark-release-recapture method can be used. The methods of measuring abundance described before work well with the plant species and non-motile or very slow-moving animal species. But they won't work well with motile organisms. Different methods are required for estimating the number of individuals in population of mobile animals. One of these methods is Mark-release-recapture method. For a single species in the area, the first large sample is taken. As many individuals as possible are caught, counted, and marked in a way that won't affect their survival. Then they are returned to their habitat and allowed to randomly mix with the rest of the population. When a sufficient amount of time has passed, another large sample is captured. Now, the number of marked and unmarked individuals within the sample are counted. In this case, the proportion of marked to unmarked individuals is used to calculate the estimate of the population size. Using this formula, estimated population size equals to the total number of individuals in the first sample multiplied by the total number of individuals in the second sample, all divided by the number of marked individuals recaptured. Now, this technique relies on a number of assumptions. First, the proportion of marked to unmarked individuals in the second sample is the same as the proportion of marked to unmarked individuals in the population as a whole. The marked individuals released from the first sample distribute themselves evenly among the remainder of the population, and they have sufficient time to do so. They also assume that the population has a definite boundary so that there is no immigration into or immigration out of the population. Also, they assume that there is few death and birth within the population. And finally, they assume that the mark or the label is not lost or rubbed off during the investigation.